DC48 Automated Sample Preparation System. Automatic decap, recap, and transport samples. Process 48 samples at the same time. Adopt with the 32-48-96 nucleic acid extraction system to realize high-throughput product transfer and PCR system establishment. Plug in the power cable. Turn on the power switch on the back of the instrument. Press the standby button in the front. Software starts automatically. Enter the password admin. Instrument self-check. At the software interface, turn on the cabin lights. The barcode scanner is on the left of the cabin. The drip tray prevents contamination from cap drip. Place up to 48 tubes at the sample area. Place protease, PCR reagents, and other quality control reagents in the last rack. Place up to 3 pieces of 96 deep well plates. Place 1000 ml tips, 1000 ml or 50 ml tips, and 8 strip PCR reaction tubes in sequence from inside to outside. Remind the needle should face forward. The waste bin for tips. Click System. At State tab, after ensure normal system status, start experiment. Sample preparation procedures. Place the sampling tubes and 2 boxes of 1000 ml tips. Place protease K in the reagent rack depending on program settings. Position 15 is default. Place standard and QC control at position 1 to 7. Place deep well plates. After all the reagents and consumables are set down, click System. At Settings tab, select 16 or 96 text per run depending on the deep well plate type. At Sample Tube Type sub-tab, choose or edit the type of collection tubes and click Save. Back to Test. At Preparation tab, select Test Reagent section. Click the number on the left to edit the sample quantity. Click the number of sample quantity. Click any number to cancel the sample addition at this position. The total of sample plus standard and quality control should not exceed 48. Click OK to save. At Mode Step section, check Load Sample. Check Load PK according to experiment requirements. Select Start position either column 1 or column 7 when the deep well plate type is 96T. At Reagents and Consumables section, click ST and QC to set processing order. Click Tip Area to ensure the tips are enough. Click Start. The experiment begins. At Status tab, the experimental status is displayed in real time. Decapping one by one to prevent cross-contamination. The pipette detects and follows the liquid level. Alarm invalid aspiration and clogged needle in time. The drip tray under the pipette effectively prevents cross-contamination. The instrument beeps and pops up notification when experiment ends. Take out the deep well plate for nucleic acid extraction. PCR system establishment procedures. Place the deep well plate with extracted nucleic acid, 1000 ml tips, 50 ml tips, and 8 strip PCR tubes in the instrument. Place PCR reaction reagents in the reagent rack depending on program settings. Position 16 is default. At Preparation tab, click System Building sub-tab to establish PCR system. At Test Reagent section, confirm sample quantity. The total of sample plus standard and quality control should not exceed 48. At Mode Step section, check Mix Dis and Product Dis. Select Start position either column 1 or column 7. At Reagent and Consumables section, click ST and QC to set processing order. Click Tip Area to ensure the tips are enough. Click Start. The experiment begins. At Status tab, the experimental status is displayed in real time. When the experiment is running, opening the door will pause the program and pop up warning. Close the door to continue the program. Pull out the waste bin of tips. The program also will pause and pop up warning. The instrument beeps and pops up notification when experiment ends. At the Result tab, click Detail to view the experiment history. Click System at Maintain tab. Set UV light for sterilization. Both the cabin and waste bin has UV light. Click Power to shut down the instrument. The standby button will be automatically locked. Disabled power on and off function prevents accidental shutdown caused by mistouch. Reagent settings. Click Config to edit reagent configuration. Click New. At Reagent Info window, input reagent name. Select S to set sampling volume. Usually the range is 100 to 300 ml. Select Elution to set the volume and well position of the elution solution. When the deep well plate type is 16T for PCR system establishment, extracted nucleic acid will be automatically transferred from the set well position of elution. Select PD to set the volume of extracted nucleic acid will be transferred. Select Retention S to set the retention volume of extracted nucleic acid. Skip this step if no retention needed. Select PK to set the volume of protease K to be added. Select Premix to set the volume of PCR reagents. Click OK to save. Choose the type of deep well plate. The default deep well plate 1 is round bottom, and deep well plate 2 is V bottom. Check ST and QC participate in extraction to proceed quality control at the same time. Sample tube parameters management. Insert the USB drive saved sample tube's parameter data. Click System, Maintain tab, and Sample tube subtab in order. Click Import and select the data file. Click Open, and a notification window will prompt successful import.